When you hear the name Chernobyl, what immediately springs to mind? For most of us, it's disaster, right? Desolation.
Yeah, a place scarred by invisible poisons. Somewhere you just wouldn't expect life to, well, thrive.
Exactly. But the research we're diving into today, it reveals something completely counterintuitive. Life, specifically fungal life, isn't just surviving there.
It's flourishing right in the most dangerous parts. It's pretty astonishing.
That's the paradox we're exploring today.
We're talking about organisms that have set up shop inside the actual damaged nuclear reactor, specifically the fourth unit shelter at Chernobyl.
And our guide for this deep dive is a really fascinating scientific study. It's called Fungi from Chernobyl, Microbiota of the Inner Regions of the Containment Structures of the Damaged Nuclear Reactor.
Right. Published back in 1999. Comes from researchers at the Institute of Microbiology and Virology, Ukrainian National Academy of Sciences.
And what's really key is where they did their work. They took samples inside the shelter between 1997 and 1998, so more than a decade after the accident itself.
Yeah, deep inside. So our mission today is pretty clear. Unpack what these scientists found. What does it tell us about life in, truly extreme environments?
And what surprising insights does it offer about just how resilient nature can be?
Exactly. But first, maybe we should set the scene. Really understand the environment these fungi call home. Because it's unique.
Absolutely essential.
Yeah.
So let's rewind briefly. April 26, 1986, the fourth reactor at Chernobyl, catastrophically destroyed.
It was during an experiment, right. Improperly supervised. They turned off the cooling system.
Yep. Led to an uncontrolled nuclear reaction.
Yeah.
And then this massive steam explosion. It literally blew the protective covering off the reactor core.
Unleashing something like 100 million curies of radionuclides just flung into the atmosphere.
And that wasn't all. The intense heat inside caused the nuclear fuel, the reactor parts, building materials. It all melted together.
Forming that highly radioactive substance they call lava. It still contains maybe 5 to 10% of the original irradiated fuel.
And it's still the main source of contamination there today.
Right. So in the aftermath, by autumn 86, they built this huge structure over the damaged reactor, the shelter.
The idea was containment. But, and this is critical, the study points out the construction standards. They weren't really up to long-term containment, not for the estimated 20 million curies still inside.
It was a fix, but maybe not a permanent one.
So picture the scientists going in there between May 97 and November 98, where they were sampling. It was incredibly dangerous.
Yeah, the contamination levels varied wildly. You had potent alpha and beta emitters like plutonium-238, 239, 240, americium-241, curium-244. Nasty stuff.
And the gamma radiation. All over the place, sometimes relatively weak, 1.5 to 25 million radians per hour.
Which is still pretty high for normal background levels.
Oh, definitely. But then you had severe levels, 40 to 220 million radians per hour.
Lethal doses very quickly for humans.
And they even sampled water that had leached through the fuel materials.
Right.
Extremely high alpha activity, 260 becquerels per kilogram. And beta activity, 10 million becquerels per kilogram.
Unbelievably hostile conditions.
And yet, what did they find?
That's the amazing part. Despite all that, they found extensive fungal growth everywhere.
You mean like visible?
Yep. On walls, ceilings, access ladders, inside cable passages, sometimes covering areas up to a square meter.
Wow. Life finding a way, indeed. And not just one type.
No, incredibly diverse. They identified 37 different species of micromycetes, tiny fungi from 19 different genera.
And the study mentioned mitosporic fungi predominated.
That's right. Those are fungi that mainly reproduce asexually, so they can colonize quickly, adapt fast. Makes sense in that kind of environment.
Were there any particular stars of the show?
Yeah, you could say that. 2 species really stood out. Cladosporium spherospermum, they found that one almost everywhere they looked.
Okay.
And Penicillium hirsutum, isolated from nearly 89% of the locations.
So those two were really dominant.
Very much so. But others were common too, like Alternaria alternata, Aureobasidium pullulans, Aspergillus versicolor, Acremonium stictum, and another Cladosporium herbarum.
It's quite a list. And didn't they find some species completely new to Ukraine?
They did, which really highlights how unique this environment is. Penicillium ingelheimans, Phelophora mellini, Doratomyces staminitis, and Sedoia polyspora, never recorded in Ukraine before being found inside the shelter.
It's like a bizarre radioactive lost world in there.
Kind of, yeah. And this leads us to maybe the most fascinating part of the study.
Which is?
The melanin connection. They found this really striking correlation. The fungi with melanin, the dark pigmented ones, they absolutely dominated in the most radioactive spots.
Melanin, like the pigment in our skin.
Exactly, same basic type of pigment. And the evidence for this being an adaptation was pretty strong.
How so?
Well, look at the numbers. The frequency of these dark fungi jumped dramatically. In most areas, from about 30% in the spring up to 100% by autumn.
100%.
Yep. Even in the weakly contaminated sites, their presence went from 17% up to 63%.
So it wasn't just chance. This suggests the melanin is doing something important.
The researchers think so. The idea is that melanin doesn't just help them survive the radiation. It might actually allow them to grow actively for long periods under these conditions.
That's incredible. Has this been seen anywhere else?
There are similar observations, yeah. Dark pigmented fungi were noted after the nuclear tests at Bikini Atoll, so it seems to be a pattern.
Okay, let's talk about the sheer level of radiation these fungi are dealing with. You mentioned calculations.
Right. The estimates suggest they're absorbing a minimum of hundreds of sieverts every year.
Hundreds of sieverts. Annually. How does that compare to, say, background radiation or human limits?
It's almost impossible to wrap your head around. It's at least 100,000 times the natural background radiation we all experience, which is usually, 1 to 3 millisieverts per year?
Okay, 100,000 times. And for humans?
For us, a short-term dose of just 2 to 10 sieverts is incredibly dangerous. Severe radiation sickness, increasingly likely to be fatal.
And the limit for nuclear workers?
It's capped at 20 millisieverts per year, averaged over 5 years. So these fungi are enduring radiation levels that are just astronomical by human standards, truly unsurvivable for us.
It really highlights their extreme resilience.
Yeah.
But what's maybe even more surprising, you said despite this, there was still decent biodiversity.
Yeah, that was another key finding. The overall environment inside the shelter maintained, as the study put it, a reasonably high level of species diversity.
How do they measure that?
They used a biodiversity coefficient. The average was 2.94, which for such an extreme place is pretty remarkable. It suggests, that life finds ways to diversify even under immense stress.
Was it diverse everywhere?
Mostly. The exception was the cable passages. Lower biodiversity there, a coefficient around 1.02. Probably because they were more enclosed, harder for different spores to get in and establish.
And these weren't just isolated species, right? They were forming communities.
Exactly. They observed fungi growing closely together in associations. Like Oreobasidium pullulans often grew right next to Cladosporium sphaerospermum.
So they weren't just competing, maybe interacting.
Seems likely. And in those cable passages, they found more complex groups like Aspergillus versicolor, Chrysosporium panorum, Stachybotrys chartarum, and Penicillium chrysogenum all living together, functioning microbial ecosystems, essentially.
Okay, so these fungi are incredibly tough survivors. But there's a practical side to this, isn't there? A potential downside.
There is. The study labels them potentially active biodestructors, meaning they could actually be breaking down the materials they're living on, even the highly radioactive stuff.
Which includes the shelter structure itself.
Potentially, yes. And that raises a really serious question about the long-term stability of the shelter. We already know its construction wasn't exactly top tier for permanent containment.
So these fungi, just by surviving and thriving, could actually be weakening the structure meant to hold back the radiation.
That's the concern. We know fungi can damage concrete and similar materials in normal environments. There's research, especially from the former USSR, showing they can reduce surface resistance, create carbonized layers, increase organic content through their acidic byproducts.
Real, measurable damage.
Right. Now, the Chernobyl environment is different, maybe fewer nutrients than some other places where fungi cause damage. The study notes differences in the types of fungi found, but the basic principle of biodestruction. It's definitely something to consider for the shelter's future.
It adds another layer of complexity to the whole Chernobyl situation. So this ecological puzzle, where did these super fungi actually come from?
Well, most of the species they identified are pretty common types found elsewhere. Soil fungi, things that break down dead plants. Some can even be mild plant pathogens.
So they weren't, created by the radiation?
Doesn't seem like it. The leading theory is simple spore dispersal. Air currents blowing through gaps in the shelter carried fungal spores in from the outside environment, just like happens everywhere else.
Okay, so spores from common fungi blew in.
And then the incredibly unique high radiation conditions inside the shelter acted like a very intense filter, an ecological selection process.
Ah, so it selected for the ones that could handle it.
Precisely. It favored the subpopulation of spores that already had traits allowing them to survive and even thrive in that environment.
And that brings us back to the melanin. Those dark pigmented species had a pre-existing advantage.
Exactly. They were better equipped from the start, and under that extreme selective pressure, they became the dominant players. It's a powerful example of natural selection in action in a place you'd least expect it.
What an incredible journey into Chernobyl's hidden fungal world. We've seen life not just clinging on, but actively flourishing in perhaps the most hostile place on Earth.
It really pushes our understanding of life's limits, doesn't it?
It absolutely does. And it has real implications, thinking about the integrity of structures in other contaminated zones.
Sure. It makes you ask, if these organisms can adapt like this under that much radiation, what else could they teach us?
Could digging into their genetics, understanding how they're modified, maybe give us clues for bioremediation, using fungi to clean up radioactive waste?
It's a possibility researchers are exploring, or even thinking further out. Understanding life in extreme radiation could inform our search for life beyond Earth in harsh cosmic environments.
The thought, isn't it? These tiny, resilient fungi inside a ruined reactor potentially holding keys to cleaning our planet, or even understanding life in space. It just shows how much we still have to learn about the sheer tenacity of life.